It's really my honor and pleasure to introduce Dr. Cori Bargmann. She is the president of science at the Jan Zuckerberg Initiative right now. She's the Torsten N. Wiesel Professor at the Rockefeller University. She's the head of the Lulu and Anthony Wang Laboratory of Neural Circuits and Behavior. She has a Bachelor of Science in Biochemistry from the University of Georgia, PhD from MIT in Biology, and did postdoctoral research at MIT as well. She spent a number of years at the University of California, San Francisco in the departments of anatomy and biochemistry and biophysics. Went through the ranks, became full professor, vice chair. Since 2004, she's been at the Rockefeller University, as I indicated, and she was a Howard Hughes Medical Institute investigator for a while, like 21 years, so congratulations on that. Like I said, she's now at the Chan Zuckerberg Initiative. She has a tremendous list of honors and awards. I'll just mention a few. She has the NIH Director's Award for Scientific Leadership because of the Brain Initiative, which she was instrumental in. Breakthrough Science, the Breakthrough Prize in Life Sciences, the Cavalli Prize in Neuroscience, the Norwegian Academy of Science and Letters, American Philosophical Society, National Academy of Science, and won the Richard Lounsbury Award in Biology and Medicine from the National Academy of Science. A fellow of the American Association for the Advancement of Science, Kamali International Prize for Research in the field of basic and clinical neurosciences, and a fellow of the American Academy of Arts and Sciences, among others. So it's very impressive. She's on editorial boards of Neuron and Cell and a number of other really prestigious journals. Huge long list of advisory board external service for prizes, for foundations, for centers, for funding agencies. So a huge number of publications with 37,000 citations and an H-index of 102. So we are very privileged today to have Cori Bargmann as our speaker. So please. And before that, we're giving her a book of photographs of the Cleveland area, which we'll send to you, as I told you before, and another little reminder. So I'll set these aside, and we'll do that later. So please, Cori.
So I'd like to start by thanking Bob for the invitation, for setting up my schedule and accompanying today throughout the different institutions and interacting with different faculty here at CASE and at the Cleveland Clinic. It's a really stimulating and exciting group of people that I relate to based on my long-lasting identity as a neuroscientist and my current identity in trying to think more about medical problems. And in my talk today, with Bob's permission, I'm actually going to cover two topics. In my talk today, with Bob's permission, I'm going to cover two topics. I'm going to talk first about work in my laboratory that I think is, while we are very much basic scientists and we study a model organism in a lot of sort of molecular detail, I think it's a topic and there are implications that could be of interest to those of you here who think about topics like neuromodulation and electrical stimulation of the brain and how different kinds of interventions might be effectively used and augmented in modifying brain function. That's the first part of the talk. And then in the second part of the talk, I'll talk about what I've been spending a lot of my time doing for the past 2.5 years, which is setting up a new biomedical scientific philanthropy in partnership with Mark Zuckerberg and Priscilla Chan. And I'm really interested in sort of presenting that to scientists that I respect and also hearing from you all what you think will be good approaches for us to take and good ways for us to move forward. So thank you for that. So I'm going to start by talking about a class of signaling molecules in the brain, which are the neuromodulators that work through G-protein coupled receptors. So these include small molecules like dopamine and serotonin, and they include a variety of different short peptides, neuropeptides. They're typically made by nuclei deep within the brain and that have projections to many different parts of the brain. And they are strongly implicated not so much in the very rapid millisecond level propagation of information between neurons in the brain as in longer scale integration of information across different brain regions. So these are molecules that are implicated in the control of motivation and emotion. They're very strongly implicated through genetics and pharmacology in the regulation of processes like sleep and hunger, mood, arousal, social drive, pain, and stress. And perhaps the most eloquent statement about their importance in the function of the brain is the fact that almost all psychiatric drugs and almost all drugs of abuse mediate their effects by acting through these modulatory systems and their related G-protein coupled receptors. And that really tells you that these things have effects on human cognition and human behavior, and even in some cases on human motor function, as in the case of dopamine. So these seem like molecules that are really important to understand. We have some amount of insight into what they're doing, but in fact there are many holes in our understanding of their underlying biology. How do I work this? Okay, good. So here's the kind of question. We have on the one hand the illustration that I used from the previous slide that identifies the nuclei that make these transmitters, their nature, and their overall effects on things like motivational and emotional states. And on the other hand, we have really detailed information about their molecular mechanisms. For example, at synapses, the G-protein coupled receptors often act in parallel to the classical fast neurotransmitters that act through ligand-gated ion channels and modify their excitability and other properties. But there's a really big gap between that level of understanding and this level of understanding. And the gap that I think of, the way that I think about it is we want to understand what the structure is of these neuromodulatory circuits. Now I know here, when people say neuromodulation, they often mean electrical stimulation of a human brain. When I say neuromodulation, I mean these chemical processes. My use is older, so I'm going to stick to it. But just to not be too irritating, I'll just get that up front. And the question is, how do they work? So for example, what kinds of distances do they work over? In terms, there was a kind of a theory that these things were sprinkler systems that sort of bathed the whole gene and bathe the whole brain in a transmitter, versus some of these very localized synaptic ideas. What time scales are they relevant over? What's the time scale of their production and retrieval? What is their relationship to the underlying synaptic circuits? One of the reasons these things are not so well understood is that they aren't associated with the classical specializations of synapses that you might see in an electron microscope, like a postsynaptic density. So it's not really known when you see one of these molecules being released where the relevant receptors are that are responding to that molecule to give rise to some kind of an outcome. And similarly, since they don't cause neurons to fire action potentials necessarily, but rather lead to biochemical changes mediated through the G-protein coupled pathways, it's not so easy to measure exactly when they've been active and where and for how long the outcome of the synapse and the circuit is being modified. So these are questions that are clearly interesting and valuable. The question is how to go about studying them. And I study them despite my interest, like all neuroscientists, in human behavior in a much simpler animal, and that is the nematode worm Caenorhabditis elegans. And the reason that I study this tiny animal is that it has a nervous system that really enables an exceptional amount of resolution into biological processes and into the functioning of molecules in neurons, of neurons in circuits, and in groups of neurons in generating behavior. This is illustrated as shown here on this slide, where the 302 neurons of the C. elegans nervous system, its entire nervous system, can be shown on a single slide. Each of these neurons has a particular name. This arrangement happens to involve sensory neurons being mostly in red and mostly up at the top, the information flow being through a set of integrating neurons in blue in the middle, and then flowing out through different motor systems in green at the bottom. There's an enteric nervous system off to the side there that's only very slightly connected to the rest of the nervous system. And these thin gray lines illustrate the synaptic connections between these neurons that have been defined from serial section electron microscopy of the entire worm's nervous system by John White and his colleagues. And so we can say that for the animal that they examine most closely, there are 6,393 chemical synapses among these 302 neurons, along with 890 gap junctions. And interestingly, in C. elegans, as in mammals, there are a lot of neuromodulators. And one of the things about the C. elegans nervous system and about nervous systems in general that came as a surprise, even to those of us who work on invertebrates, is how incredibly conserved the molecules are that are used to generate nervous system development and function across different animals. So most of the molecules that are present in the mammalian brain and in the human brain are present in the brain of C. elegans. In fact, almost all of the genes that can mutate to give rise to diseases like autism or intellectual disability disorders are conserved between C. elegans and mammals. So this is not to say that C. elegans has autism or anything that similar to that. Thank you, Makesh. But it is to say that the basic building blocks that are used to assemble a nervous system are present in recognizable form across very different animals. And so if you're trying to get at sort of fundamental questions like the ones that I raised on the previous slide about molecules like dopamine or serotonin or neuropeptides, you can answer them in these questions, in these systems, and then try to take those conclusions and see where they might or might not apply to more complex nervous systems. And I'll try and close the circle on that toward the end of the neuroscience part of my talk by telling you some places where I think there might be interesting ideas that emerge from this that could be tested more broadly. So we have this C. elegans wiring diagram. There are other really attractive things about C. elegans. One of them is that it grows really quickly. A generation time from egg to egg is just over 3 days. The animal can grow as a self-fertilizing hermaphrodite, which means that you can get hundreds or thousands of genetically identical animals in a very short period of time if you want to rigorously control their outcome. They're also susceptible to classical genetics, which lets us make hundreds of thousands of animals that differ from the wild type by a single genetic mutation, and allow us to examine those in a lot of detail. And so, and they're also optically transparent, which allows us to manipulate their nervous system using tools like optogenetics, for example, very easily, or using things like genetically encoded calcium indicators in live animals, viewing them through a microscope, and then observing the activity of the nervous system. So this has enabled us to study a number of different systems. And there's sort of a question here, which is the wiring diagram was published over 30 years ago. I have been working on C. elegans continuously during that time, and so have a lot of other people. So why haven't we solved the wiring diagram? Why couldn't I take that drawing from the previous slide and tell you exactly how information would flow through that nervous system to give rise to behavior? And the answer to that turns out to be that the wiring diagram is ambiguous, that we don't think that there is a single way to read the wiring diagram, but rather that its abilities can be changed in a dynamic fashion by the presence of neuromodulators that alter information flow. And I'm going to give you what I think is the simplest and clearest example of that right now, based on work that Hyun Jang, a graduate student in my lab, did in collaboration with Piali Sengupta's lab at Brandeis University. And that was to look at the response to pheromones, to social odors that C. elegans to detect and respond to. And it responds to those in a context-dependent manner, and actually in a way that depends on naturally arising mutations in the wild populations that enable different animals to behave differently from one another. So sometimes the animal is attracted to pheromones, and sometimes it's repelled by pheromones. And it turns out that in both cases, the pheromones are sensed by exactly the same olfactory neurons, which I can pick off of that wiring diagram. And one of the beautiful things about this organism is that we have a community that works on it. And when I point at this neuron, I know exactly where that neuron is. I know what its connections are. Anyone else who's working on this organism can point at the same neuron and know that they're studying the same thing, and that the genes expressed there will be the same, and so forth. So we're in a great position to build on each other's work through this. And so using the wiring diagram and using our own work and collaborating with a chemist, and then thinking about this wiring diagram, we realized that the same neurons detect the pheromones, whether the animals are repelled by it or attracted to it. And that what happens is the information flows down one of two alternative downstream pathways. So sort of at rest, The basal rate of activity represents strong signaling through a gap junction connection and a gap junction circuit that leads to attraction or aggregation. So animals gather into these little piles of worms here. However, in different contexts or in different genetic backgrounds, a neuropeptide receptor becomes activated. This neuropeptide receptor is expressed not on the olfactory neuron, but on one of the targets. The sources of the peptides that related are expressed in yet a third location. And when this peptide receptor is active, it shuts off the gap junction circuit at a downstream stage and therefore eliminates the attraction behavior that uncovers the ability of the same neuron's chemical synapses to mediate a repulsion from the pheromones, and that causes the animals to scatter. So this clear example gives us a sense of what it is that we were missing in the wiring diagram, which is that it was both ambiguous, encoding two behaviors, and incomplete. It wasn't showing us the roles of these neuromodulators in modifying those behaviors. But it's good from the perspective of the animal because it makes the animal able to rapidly switch its behaviors back and forth between these different kinds of outcomes based on the situation it finds itself in. So we think that the important conclusion from this, that comes from this and from other work in my lab and others, is that the role of these neuropeptide signals and neuromodulatory signals is to select among the various connections in the nervous system to reroute information flow and allow different behaviors to take place. And so, for example, it's easy to think about how your own response to olfactory cues is different, whether you are hungry or not hungry or suffering from the flu, that the sort of immediate response to those might lead to a strong arousal response, an aesthetic appreciation, or a wave of nausea immediately after the arrival of the same olfactory stimulus, not because the sensory neurons were any different, but because the information flow was under the control of cues related to your physiological states that helped you to interpret them quickly with respect to those physiological states. So this is a starting point for the work that I'll be talking to you about. One of the things that's important about that conclusion is that it relates the animal's behavior to an internal state. So instead of thinking about a behavior as something that's necessarily just driven by a particular sensory cue, we think of a behavior as something that results from the interaction of sensory information with an internal state of the animal that's defined by the use of these neuropeptides and neuromodulators. And so in order to study that more closely, we've actually taken a step away from the sensory cues and taken a step back to look just at the spontaneous behavior of the animal in uniform environments, because we think that's a really good way to evaluate quantitatively what an internal state is and how it's represented. So we're just stripping the sensory information out of the picture by making that uniform under all circumstances, and then asking, for example, what do animals do, and can we use those behaviors as a way of getting readouts of internal states? And so one of the behaviors that we started studying, and we in this case was Steve Flavell when he was a postdoc in my laboratory. He moved a couple years ago to MIT, where he now has his own group, is a set of behaviors where animals are just sort of spontaneously moving across a bacterial lawn, which is what they eat, and then giving rise to different sorts of relatively long-lasting behavioral states. And these behavioral states are characterized by different locomotion patterns. There's a state called dwelling, where animals stay in a small area and eat the food and move relatively little. They turn very often, so they have a high angular speed and a low velocity. And this is characteristic of this behavior state, and typically a dwelling state might last for about 8 minutes at a time. And then all of a sudden, for no reason that we can see, the animal will take off from that location and race around the plate for a couple of minutes and then go to a different location and settle down. And while it's roaming, it's moving very quickly, and it doesn't do many reversals, so it covers a large area in a short period of time. And here's the tape of a videotape of an animal, single animal, for 90 minutes, showing it moving back and forth between long dwelling states and long roaming states on a completely irregular schedule. This is a very typical recording of this kind of behavior. So this kind of behavior is observed in all kinds of animals. You see it in foraging animals in field studies and neuroethology. You see it in Drosophila, where this is called roving and sitting instead of roaming and dwelling. Once you get into it, you see it everywhere. You see it in toddlers on a playground. And it's an example of a long-lasting, internally generated behavior. There's something about the internal state of these animals that's different across these different time periods. And what is that? And so what Steve did was to do a systematic study of different kinds of molecules that act in the nervous system to ask which were important for roaming and dwelling. And after a while, he emerged with 2 different molecules that he thought had particularly interesting roles in these behaviors. And those 2 molecules are the small molecule neuromodulator serotonin, well known to us as the target of drugs like antidepressants as well as LSD, and a short neuropeptide called pigment dispersing factor in C. elegans, or PDF. In mammals, this is called vasoactive intestinal peptide, or VIP. So this is one of these conserved neuropeptides I was telling you about. And what caught Steve's imagination about these particular molecules, as evidenced by mutations either in the molecules themselves or the enzymes that produce them, or in particular receptors that detected them, is that in these animals, both roaming states and dwelling states were present. In these animals, roaming states had few turns and fast speed, dwelling states had many turns and a slow speed. The individual actions were all fine, but the durations were altered. So in animals defective in serotonin, dwelling states were much shorter than they were in the wild type, whereas roaming states were much longer than they were in the wild type. And conversely, in animals defective in PDF, dwelling states were greatly lengthened, but when the animal roamed, which it never did for more than about a minute at a time before settling down again, its roaming looked perfectly normal. And so these are molecules that seem to be involved in setting the duration of roaming and dwelling states, not their substructure. And this is what we were looking for in terms of thinking about what it was that generates a long-lasting, motivated behavioral state. So just to kind of summarize this, and also the effects of some double mutants affecting both the transmitters and their receptors, what Steve's work suggests is that these 2 molecules have an antagonistic relationship, that each of them stabilizes one of these 2 behavioral states while actually destabilizing the opposite behavioral states, and extends the roaming or dwelling states from relatively short time periods of a minute or so to much longer time periods. So this is our first idea about what modulators are doing. Now we move on to the next great thing about C. elegans, which is that we have this modulator or modulators, we have their receptors, and we have a wiring diagram. And furthermore, from previous work from lots of people, we actually have here a compressed version of the wiring diagram that represents the foraging circuit that's used to generate both roaming and dwelling related behaviors. So the same neurons overall are involved in forward locomotion and reverse locomotion and various kinds of oscillations and turns. Here's the set of those. They seem to be differently active in roaming and dwelling states, but they generate the same output behaviors. And there are exactly 7 neurons in the worm, 7 types of neurons, that make serotonin. And we know what all of them are, and we know what their connections are. And so, for example, it kind of jumps out at you right away that 3 of those 7 neurons are directly connected to neurons of the foraging circuit. And potentially then would be the regulators of serotonin. Now, there's some 4 other neurons that don't look so interesting. They're motor neurons, or in that peripheral enteric nervous system. But the joy of having a small number like this is you don't have to just kind of guess which neurons are important. You can actually test which of these neurons are important, which Steve did by using Cori-Lox recombination to knock out serotonin production from each of these neurons individually and look at the behavior. And upon looking at that, he found that the neurons that were important in this behavior were not the ones that were directly connected to the foraging circuit, which did absolutely nothing that we could tell in the spontaneous behavioral assay, but rather these neurons that were essentially outside the foraging circuit, 2 of which were critical for generating the appropriate roaming and dwelling intervals. So serotonin neurons promote dwelling. One of those is a serotonin neuron in the pharynx, another is a motor neuron. So these are not part of the normal sensory to motor neuron top to bottom wiring diagram. These neurons are not directly connected to any of the important neurons in this wiring diagram. Steve mapped the sites of action of their receptors. Their sites of action of their receptors are scattered in multiple locations throughout the foraging circuit. So the circuit is being modified by action of serotonin on multiple targets. So there's a distributed logic here with multiple sources, a sort of bottom-up or inside-out organization instead of a top-down organization, and then multiple receptors. And Steve repeated that same experiment then with PDF, the neuron set that promotes roaming behavior, and he emerged with a different specific answer, but the exact same general answer. Multiple neurons make the neuropeptide that stimulates roaming. Multiple neurons express the relevant receptor. These neurons are not directly connected in the wiring diagram, and the effects are bottom up rather than top down, again, coming from sort of outside or low down in the circuit. And even the targets they act on are largely non-overlapping with the serotonin targets. So the circuit that's regulating this behavior is scattered among multiple sources and among multiple recipients to modify the behavior to give rise to different outcomes. So what we see is a wireless and distributed circuit that is superimposed on a wiring diagram for foraging to modify its behavior. So let's learn a little more about this. Now we know exactly which serotonin neurons we should be looking at, and we can ask, well, when are they active? Are they a sprinkler system that's just dumping serotonin from the pharynx throughout the worm, or are they something a little more selective? So here, Steve used genetically encoded calcium indicators to record the activity of the serotonin-producing neuron at the same time that he was recording the behavior of the animal. And you can see, for example, there's a very sharp transition associated with the transition from a high-speed roaming state to a low-speed dwelling state. And the activity of the serotonergic neuron is tightly coupled to that transition. So this neuron becomes active exactly when the animal starts to dwell, the behavior that serotonin controls. And furthermore, here's something interesting. Most of the neurons in the circuit I showed you previously might be active for relatively short periods of time coupled to movement. But this neuron remained active for well over a minute. So its spontaneous activity included this high, long-lasting pattern of activity that is potentially related to the long-lasting duration of the dwelling behavior, although in this case it's quite a bit shorter. But still, it's minutes long. We're not used to seeing neurons, even in C. elegans, that become active for a minute or 2 minutes at a time. We're used to much shorter time periods. So we're picking out some things about these neurons that distinguish them from other neurons within the wiring diagram, and that make them interesting with respect to the behavior. And Steve could also ask how tightly the activity of this system was coupled to the behavior at a functional level rather than just watching. Is there a cause and effect relationship? And he did that using optogenetic methods to either imitate serotonin by stimulating serotonin neurons or inhibiting the neurons that express the receptor, which in this case is inhibitory. Or he could antagonize serotonin by doing the exact opposite manipulation. He could look at animals that were either roaming or dwelling, do these light stimulations, and then ask whether their behavior was modified by either imitating or antagonizing the action of the serotonin system in real time. And what he found is that if he imitated serotonin, in animals that were roaming, they shifted into a dwelling state. That's the sort of orange to blue transition state here. If he antagonized serotonin in animals that were dwelling, they shifted into a roaming state. Not instantaneously, but over a period of time, not so dissimilar from the action of those serotonin neurons that I showed you earlier. So long-lasting action of neurons leads to a slow change in behavior. And then there's also kind of a fun result, which is that if Steve took animals that were dwelling and he gave them a little pulse of serotonin, that would prolong their dwelling state. So they would now dwell for longer. And we think that's how you end up with the 8-minute overall time frame of this, is multiple pulses from this neuron. So serotonin actually is instructive. It guides the outcome of this behavior. So this is giving us one insight into timescales. Thinking about this, another postdoc who came to the lab, a postdoc named Shai Stern, who has just moved to start his own laboratory at the Technion in Israel, decided that there was no reason to stop filming animals when you would film them for 90 minutes. You might as well just film them for their whole lives. And that is a practical experiment to do in a short-lived, rapid generation time organism like C. elegans. And so Shai set up something we call the black box in the lab, because it is in fact a black box. And it's a very well-controlled environment where temperature, humidity, and illumination are tightly controlled. And you can put many animals inside this box and many small cameras, and then you can monitor them continuously from the time eggs hatch until the animals become adults, about 2.5 days later, and ask what kinds of spontaneous behaviors do they generate, not just on the time scales of minutes, like roaming and dwelling, but in this more or less uniform environment over their entire developmental history. And when Shai did that, he found that spontaneous behavior is not just structured in roaming and dwelling on the time scale of minutes. Spontaneous behavior has an extremely reliable structure that you observe over many animals across their developmental trajectories. So this chart here is a heat map. Each row, it's about 100 individuals. Each row represents a single individual tracked throughout its development. The slashes here are molts that punctuate developmental stages. They really help us keep these guys tightly synchronized. And then the colors represent the fraction of time within a 10-minute period that that animal spent roaming. And so, for example, whenever there's a red dot, that animal spent the entire 10 minutes roaming. When there's a very deep blue dot, the animal spent the entire 10 minutes dwelling. They're awake throughout this experiment. And what you can see is that there are very clear structures. For example, if you look at the third larval stage, the animals are extremely active with a lot of roaming during the first few hours of the larval stage, and then they settle down for the last half of the larval stage with relatively low periods of roaming and a lot of dwelling. If you look at the fourth larval stage, you see something different. You still see a big peak at the beginning, but then you see a second peak later in the larval stage, and so forth. And so there are these very structured outcomes that tell us that behavior is not just structured and spontaneously generated kind of from minute to minute, but actually over many hours and over the developmental time scale of the animal. And so in thinking about that, Shai went, these experiments are, they take a while. Actually, the analysis takes even a lot longer than the experiment. But he screened through a set of mutants that we thought might have interesting effects on these behaviors. And he emerged with some of the same mutants that I told you about earlier, but also with some new mutants that tell us some new and interesting things. So he looked through mutants that affect neuromodulators and their receptors. And what he found is that many of these neuromodulators affect movement over development as well. So here's serotonin that I showed you earlier. I told you earlier that serotonin does a lot of roaming. Serotonin mutants do a lot of roaming, and sure enough, they have a lot of these red colors here. And in fact, they roam more at every single stage of their life while preserving the same overall structure of the behavior. So for example, they roam a lot more in the first half of the third larval stage than in the second half. But when Shai looked at some other mutants, he saw some different effects. So here is a neuropeptide receptor mutant. These animals, like serotonin defective mutants, roam more, but they do so while also disrupting the developmental structure of the behavior. And that's easiest to see in the very newly hatched animals. So wild-type animals are very quiet when they first emerge from the eggshell. They just sort of lie around for a few hours before they get going. And you see that in the serotonin mutants too. But these neuropeptide receptor mutants, they like race out of the eggshell and like run around the plate for several hours before they start to settle down. So there's something very well defined about their early developmental behaviors that are distinct from those of the normal behavioral trajectory. And that makes us think that these are linked to something relevant about the way that these behavioral trajectories are defined. And here's another mutant that's actually more active than a wild type, specifically in the second larval stage, but much more roaming oriented at every other stage of development. So we can see these more complex and much longer lasting timescales embedded in these neuromodulatory circuits as well. And we could start to test hypotheses, for example, about the level of activity of this particular neuromodulator at different time frames that we can now test and see if that's true. So here's the conclusion to the work I've told you about neuromodulators. So what we see is that the wiring diagram of C. elegans that we know so well really is not a set of invariant instructions. It's a set of options. And the neuromodulators help the nervous system select among those options in a flexible and dynamic fashion. And we can say a set of things about these. These are orthogonal to fast circuits with distributed sources and receptors. The source neurons are interesting. They have a long-lasting activity that's measured in minutes and not in milliseconds or seconds. They seem to be able to select among the connections in the wiring diagram to strengthen some and weaken others. And in fact, G-protein coupled receptors are well known for affecting presynaptic efficacy, and therefore could be transmitter release, for example, in fast circuits, to reroute information flow. They seem to be organizing higher order coordinated circuit states that have many different actions associated with them, affecting state transitions and the duration of existing states. And finally, they operate over long timescales, minutes, hours, even days, that they can have relevant roles in behavior. And so one way of thinking about what we're doing here is we're saying, like, here's the foraging circuit that I showed you earlier with the key neurons in it. And if you just look at this circuit, maybe armed with what I just told you, it's like this circuit is really overconnected. These neurons there are just a lot of synaptic connections between different neurons within this network. And we can think of this sort of modulatory cloud as picking out a subset of these connections that are more relevant in a given behavioral state defined by a given modulator. And this, I think, is a useful principle to think about with respect to the human brain as well. So this is an image, I think, from DTI imaging of a human brain, a diffusion-based imaging method. And it just shows how incredibly interconnected different brain regions are. And I think a useful way and hypothesis to test about what these different modulatory neurons are doing is to consider the possibility that what they are doing is strengthening or weakening subsets of these different connections to change information flow in a real-time way. And the questions that we brought up in C. elegans then become testable hypotheses. For example, if you monitor the activity of these neurons that, again, act at a distance and act orthogonally to the main circuits to route information flow, are these neurons active for longer periods of time? Recent genomic work has shown that these neurons share in common a set of gene expression patterns that they do not share with the neurons like the GABAergic and glutamatergic neurons that mediate fast transmission. So there may be some interesting things there, just the molecular identity of these neurons that will help us think about the way that they're working. And finally, for the clinicians in the audience, you know better than I do that one of the characteristics of psychiatric disorders in people, and even neurological disorders like Parkinson's and Alzheimer's, is that they wax and wane, that the symptoms might be very severe one day and much milder the next, and that there's some kind of changes going on, apparently, that can affect the flow of information through this system. My mother had Alzheimer's diseases, and there would be times that she was completely checked out for a day, and then times that she would come back in and be all there and present with all of us. And so I think about these kinds of information flow. I do think very much about how things like electrical stimulation can be used to help people with these diseases, but I also think about whether these endogenous systems represent targets for potential modulation for what was going on in her mind and on her better days that we might be able to bring into this equation as well. So here's the C. elegans part. There's still lots to learn. I think a lot about how timescales are represented in the nervous system. We have a lot of questions that we still need to know. I kind of went fast and loose through some of the things about what those transmitters were actually doing to that foraging circuit, to which we don't yet know the answer. But I think we have some sort of a sense, for example, that different kinds of pieces of information might be encoded on different time scales, that when we think about these intermediate time scales, we should be thinking about these neuromodulatory molecules, and that we can consider those in the context of all the other things that we love to study, like development and learning, as a separate input to the nervous system's plasticity and flexibility. So these are the people in my lab at the Rockefeller University currently. This is Shai, who did the work on the long-term monitoring of behavior. That took a lot of analysis, which was done by a physics fellow at the Rockefeller, Christoph Kirst. There's another body of work that I didn't get into today about action potentials that several other people in the lab worked with. This point in the slide, I mentioned Steve Flavell, who's now at MIT, he and Zhang, who's now at the Buck Institute in California. And it's also traditional at times like this to thank a lot of the people who supported the work through their funding. But one of the things that occurred to me when I think about this work more and more recently is that there's a set of people who aren't thanked when you do a body of work that seem to me are also very important in the progress of that work. And I've listed here some of who those people are that have had a tremendous effect on the work in my lab over the years. So the first is that various resources and technology have been absolutely critical for the work that we did. So everything my lab did was based on the C. elegans wiring diagram, which was a back-breaking project that took 10 years of electron microscopy and reconstruction to put together. And yet my lab still turns to it, every day. I have a tattered first edition of this book in my office that we still look at to try to understand what this might do. I kind of went fast through a bunch of interesting mutants and genetic manipulations. Well, those all came out of the C. elegans genome project. It was the first animal to have its genome sequenced. And again, a lot of people worked really hard to make those reagents available, to make everything molecular that I did 10 times faster. There were tools that came into the lab from physics, like lasers. There were tools that came from engineering, the silicon chip industry, where microfluidics was really developed, then led ultimately to the return of that technology to biology. There are genetic tools, like the genetically encoded calcium indicators and the genetically encoded tools for activating or inhibiting neuronal activity. And like the calcium indicators, it took almost 20 years for them to get to the really good stage they're at today. People worked on this for a very long time to make these available to people like me and to others. And these things were critical for the work in my lab to make the progress that it made. And the other thing that is very much on my mind is how much other people that I worked with, collaborated with for many years over numerous papers, moved the work forward, taught me things I didn't know, held me to a higher standard. And that included people like Larry Abbott, who's a theoretical and computational neuroscientist, Michael Marletta, a chemical biologist, Leonid Krugliak, a population geneticist. Their very different ways of looking at scientific problems tremendously enriched the work that my lab did. And it's this kind of perspective on what makes labs move forward that was on my mind when I was approached by Mark Zuckerberg and Priscilla Chan about 2.5 years ago. Mark is known to many of you as the founder of Facebook. These are their 2 children, and on the birth of their first daughter, Max, about 3 years ago, Mark and Priscilla decided that they were going to give away the majority of their wealth in their lifetime for social purposes. So the resources come from Mark. Priscilla is a practicing pediatrician. She is the child of immigrants. And before she went to medical school, she was an elementary school teacher. And so from her own life, she has a very strong sense that she was incredibly fortunate and lucky to end up where she ended up, and that she would like to make the world a little bit so that luck was less of the equation in how successful people are. And so these are the things that very much motivate her interests, and her commitment to this kind of empowerment have been what motivate the work in the philanthropy. First of all, education, because education gives people a chance. Second, biomedical science, based on her own experience as a pediatrician, and based on the fact that health is a great equalizer. And finally, justice and opportunity, where they're doing work in immigration and criminal justice reform as part of their programs. So the philanthropy has 3 different pillars. And about 2.5 years ago, they asked me whether I would come and lead the biomedical science part of their philanthropy, to which they committed as the first tranche, $3 billion over 10 years. And I said, well, what would you like that to accomplish? And they said, we would like to support basic science and technology that will make it possible to cure, prevent, or manage all diseases by the end of the century. So there's a lot to be said about this sentence. The first is emphasis on basic science and technology. We're not going to do healthcare delivery. We're not going to do healthcare outcomes. We're not going to do electronic health records. Thank goodness. We're going to try and build something that will have an impact on the long term. We're going to try and, it's science and technology that will be making a difference in 20 years or in 50 years. And so that's the emphasis. Second thing to say about that is this kind of long time frame puts you in a different perspective about what it is you're trying to do. That many of the great workers in the scientific enterprise are focused on the next grant cycle or trying to solve a problem right now in the present, it's nice to have the flexibility to try and take a longer view of what's going on. And maybe the third and most important thing to say is think back 80 years about where medicine was and what the top killers were of people worldwide, and think about what medicine has accomplished since then. 80 years ago, people were just starting to develop the first modern antibiotics. So before then, 3 of the top 10 causes of death worldwide were bacterial diseases that we now cure with antibiotics. We did not know 80 years ago that cigarette smoking causes cancer, and so we can now prevent a lot of cancer. We did not know that high blood pressure and high cholesterol gave rise to heart disease and strokes. That was discovered based on the Framingham Heart Study in the 1960s. And that in turn led to the development of great blood pressure medications, of the statins, and then other advances from technology like stents and transplants and the ability to suppress the immune system to make those take. Cancer chemotherapy, more recently immuno-oncology, which is the advances that are being made in cancer now based on basic science and technology starting largely in the 1970s mean that death rates from cancer have been decreasing by about 1% per year in the United States every year for the past 25 years. So I think there is a lot to celebrate when we look at what can be accomplished in this kind of a time frame. And even so, I would say that maybe 1,000 years is more realistic than 80 years. But Mark's an engineer, and engineers believe they can make anything happen faster. And so we just need to make things 10 times faster. All we need to do is accelerate biomedical science by a factor of 10. And that means making everyone a better scientist. That means taking everyone who's in the system and enabling them to do better work. And what we believe, and you can tell what I was telling you about my own work in worms, I believe is the approach to that, is you make every scientist a better scientist by developing new tools and technologies, and by supporting open and collaborative models of research that let people pool their expertise to make progress more quickly. And that's our focus for the first 10 years, to see if we can make some of these changes and then see acceleration. So here are the approaches that we're taking. We are trying to find new ways to get people to collaborate across disciplines, science and engineering, also medicine, bringing in patients as participants as well. Second, enabling tools and technologies symbolized by the microscope. Before the microscope, no one knows the human body is made of cells. After the microscope, everyone can see this for themselves. What's the next microscope? And the third pillar is actually a big policy pillar of building support for science, public support for science, research support, funding support, private and public, working with others in philanthropy and in governments to try to increase, to kind of recognize that if we need science to move 10 times faster, we need more science. So that's part of our goal as well. And we have these sort of watchwords that we support: people, technology, collaboration, and open science, networking, and the sharing of information. So we're doing this by taking kind of a multiple approach that's atypical for a philanthropy. So we do some things that are very typical for a philanthropy, which is that we fund people. We have calls for applications in areas that we're interested in, and we fund grants. And the part of the organization that I'm in charge of, the science program part, is in this kind of traditional mode. But we do a second thing as well, which is that within our organization, we have a team of software engineers, product managers, people from the software industry who have been in the private sector where a lot of advances have been made in the past decade or so that have not yet returned to areas like science and medicine. And so they are now coming back to see if we can bring those advances toward the kinds of science that we all do. And finally, this is actually turns out to be a really big gap, and so you need a translation layer in between. And within our group, we have a set of computational biologists who learn on the ground, who understand both sides well enough to understand where the opportunities are, and who sort of do explorations, pilot projects, collaborations, to start to see where we can put something bigger together. And so this is who you would see if you came to our place. You would see the program team working on grant funding, the computational biology team, and a lot of software engineers, because it turns out it takes a lot of engineers to make good software, and that was something I learned when I got there. So we have to choose projects. We can't do everything. We have kind of another thing that we say is we can do anything, but we can't do everything. So what are we going to work on? So we look for advice. We're very serious about listening to the community. We have a fabulous scientific advisory board who represent people who have been, for example, directors of the NIH, or the former CEO of Genentech, or the former head of the Howard Hughes Medical Institute, and some people from technology as well, who are helping us know what's been done, identify gaps in the current scientific landscape, and evaluating our ideas to decide which ones make sense to move forward. These people are very involved in our work, and we're very grateful. And then second, this is a set of senior people with a lot of wisdom, but the people who really know what is needed to accelerate science are the scientists. And so we spend a lot of time, for example, hosting workshops where we'll bring in 20 or 30 people from a field of interest, like computational tools for microscopy, and spend 2 days with them and get them to explain to us what is going on in that field, where they would like to be, and what the best steps are to get there. So these people range from graduate students to senior faculty. There are workshops, we hold hackathons, we have meetings, we go to other people's scientific meetings, organizational meetings, we visit the NIH, we visit foundations, and we just keep asking questions. What can we do that will have a systemic effect, not for one lab or one institution or one project, but for a whole field? And where can we make a differentiated impact that's different from what other philanthropies are doing or the NIH is doing? Because the $3 billion is a lot of money over 10 years, but it's 1% of the NIH budget. And so we're not going to make a difference if we just do exactly what the NIH is doing. And so what we're doing is taking this mindset and doing some experiments and seeing how can we promote these ideas of collaboration and technology development. So like one experiment we're doing is that we're funding a collaborative research site that links together scientists and engineers in the San Francisco Bay Area, where we are also based. In this case, scientists from Stanford, UCSF, and Berkeley, 3 institutions that have a history of never working together, are able to work at this biohub, this sort of neutral site where there's a lot of really interesting technology. It's led by a professor at Stanford, Steve Quake, and a professor at UCSF, Joe DeRisi. And they're staff members who specialize in technologies. There are about 100 faculty at the partner schools who are affiliated with the biohub, and they're working on different science projects. And in addition, CTI has embedded a set of software engineers in the biohub to ask the question, is the right way to accelerate science to just drop the engineers into a scientific site and see what they can come up with? And from that, we have actually just sort of prototyped our first project that is emerging in the world. It's based on Joe DeRisi's work on pathogen detection through metagenomic sequencing. He developed some algorithms that were possible that would enable you to take just a blood sample and identify with any previous knowledge the pathogen that was causing the disease, even if it had never been previously seen. It cost about $2,000 per sample, and our engineers kind of got their paws on it and ripped it up and put it back together, and then it cost about $18 per sample. And so now we're partnering with the Bill and Melinda Gates Foundation to move this tool, IDSeq, to 10 of their sites in 10 different countries around the world to help to do global health and pathogen tracking. And in our first pilot project with the Gates Foundation, in a pediatric hospital in Dhaka, Bangladesh, there had been an outbreak of pediatric meningitis. It was poorly understood, and using IDSeq, actually, the Bangladesh group were able to show that there was a previously unknown variant of chikungunya virus, a mosquito-borne virus that was causing meningitis in these children. So this is a good way of potentially increasing our knowledge of diseases at a scalable and affordable mechanism across different sites. And it's a great collaboration of the brilliant scientist, the engineers who kind of make it scalable, and then this amazing ground game from the Gates Foundation in infectious disease that lets this come together. We're also working a lot on tools because we believe that we want tools. And we want tools that are robust and reliable and scalable and shareable. We want them to have the broadest possible impact. And we're viewing that very, very broadly. So part of that is, again, more in the area of funding, part of it's in the area of building. We're funding tools in the open science environment, like tools for protocol sharing and the bioRxiv preprint server. We're building, in this case, a software tool that uses predictive, personalized methods to identify papers that will be interesting to you from the scientific literature, using artificial intelligence and machine learning. So this is a tool to enhance knowledge sharing and discovery, or a set of tools. Another tool that we're building is a much more biological tool. We're contributing to the formation of something called the Human Cell Atlas, which is a scientist-led international open-door project to build an atlas of all 30 trillion cells in the human body. We've known for a long time that there are a lot of cells in the human body. We don't know exactly how many there are. Advances in single-cell techniques and computational techniques over the past 5 years or so have made it seem reasonable to actually figure out how many cell types there are, what genes they express, how many of each, how stable their states are, and so forth. And this is being led by Aviv Regev and Sarah Teichmann. It's scientist-led and scientist-run, and we are providing support for helping this project get off the ground. And this to us is an ideal of the sort of solve all diseases. Every disease that affects humans has a cellular basis. Some cells in the body are not doing what they're supposed to do, or they're doing what they're not supposed to do. So if you have the equivalent of a genome project for cells, you will create a foundation that people can use to study every disease. And that's an example of something we're thinking about. What Mark and Priscilla said led us in a direction that might not have been the first direction otherwise. And again, we're taking a multi-pronged approach. Our software engineers are working on a cloud-based data platform for putting all of this information together in a sensible way, in collaboration with some of the great groups that work on data platforms in biology, like EBI and the Broad. And we're funding wet lab work and computational work in labs around the world, developing technology, building computational tools. And right now we're starting grants to build the first draft of the atlas with different tissues. We're funding over 200 groups in 15 countries to do this work. So we have other things that are going on. We've just started our first explicitly disease-based project. Again, it's very much about a class of diseases, not one disease. We want to get people who work on neurodegeneration together to ask whether there are themes that might apply across multiple diseases. We also want to bring some new people and ideas into these very challenging disease fields where it's been very hard to make progress. And we want to do this in particular with interdisciplinary teams and younger scientists. So again, focus on basic science, focus on mechanisms in cell biology, not trying to take shots on goal right now and figure out cures right away. What is going on between a genetic risk factor for Alzheimer's and the death of the brain? Well, it's something in here with all these cells. And so what can we figure out? And so we've just funded 48 groups to work on this. One of our programs is actually named after the late Ben Barres, a visionary scientist who was both a great advocate of young scientists and of women and minorities, but also someone who thought about brain disease in a different way and was someone who was championing the role of glia in the brain long before everybody else figured this out a couple years ago. And finally, we're starting some other projects. We're just starting a project in imaging and microscopy, where we thought about what it, again, in conversation with many people about these fields and how critical they are for modern biology, we realized that a really under-supported and important group of people in this field are the staff scientists who work at core facilities, who make innovations, who disseminate new technologies, who educate your lab members. And so we are starting to work with a group of them to start to create a stronger infrastructure for this community. We're supporting open source software projects in imaging. And our very newest project, announced just this week, is if we're going to have more support for science and we want more science, we need more people in science. And we believe that there's a good reason to push for more diversity in science education. The school that has the best reputation for bringing underrepresented minorities into sciences is the University of Maryland, Baltimore County. And we are working with them, UC Berkeley and UCSD, to see if we can clone their very successful program at some of these UC schools to try to bring more underrepresented minorities into science, starting at the undergraduate level. So you can see, we care about people, we care about technology, we care about collaboration at every level between institutions, between individuals, and we care about opening science up, for example, in open software projects. And we get people together a lot. We want them to share their code and their protocols and their preprints. We think that's part of the key of all of this. This is a real photograph of real people writing real code. And we think if we're going to do a new philanthropy and we're going to try and do something different, we also have to have a new way of thinking about what success is. And I just want to say that, in science we have these specific measures of success. For example, publications in pure basic science are it. And we just don't think that we should be thinking just about that. We should be willing to publish, we should be willing to fund someone who just generated incredibly valuable data sets or protocols or software. We should evaluate work based on not just did you do something good with your mouse, but did other people do good things with your mouse? You should get as much credit then as when you did the work. And sort of requests and reuse, public repositories is part of our funding mission. And finally, we want to reward collaborative, if we say we're in favor of collaboration, we should reward people, not just if they're the senior author of the paper, but if they contribute in important ways to the work that gets done. So just sort of thinking about these things in different ways, we will need to do that in cooperation with institutions like yours. You all give the jobs to people that we are ultimately going to support. And so I think in a medical setting, that is actually better understood than it is perhaps in some basic science settings. I think there's a lot to think about, but I'm really interested in your feedback on where you think that these things could break down. And so finally, just to tell you, you can look up everything we're doing on the web. We just recently published all our grants. We're looking for people if you're interested in joining us, and I thank you for your time.
Questions? So, in your opening figure of the brain with the neuromodulator circuits, I couldn't help but notice that the slide projected a bit of cerebral chauvinism. The cerebellum was not indicated as being part of any of those circuits. And it's actually pretty clear now that the cerebellum can affect behavior and cognition and emotion. And I'm wondering if, one, are you thinking about that using your cerebrum?
Or my cerebellum.
And two, when you study something, a model organism like C. elegans, does that really have a cerebellar equivalent?
So the answer to the first question is yes, I agree that the cerebellum is important, and I apologize that it was not included. I also believe that the spinal cord is really important, and that's not on there either. And in fact, when we were doing the brain initiative, I called a number of people in neurology and psychiatry and things like that and said, what do you need? It's sort of the same process. And the neurologist said, just don't make this all be about the cortex. They're like, neuroscientists are all about the cortex, but let me tell you, that's not the problems we see. The brainstem, the spinal cord, the peripheral nerves, pain syndromes, and so forth. So just to say, I'm with you there. And do non-vertebrates have a cerebellum? We don't know the answer to that in general, whether the cell types have a deep evolutionary relationship. And I think that that's the kind of thing that will emerge from genetics and molecular characterization. If you end up with a cell type that expresses the relevant transcription factors and channels and plays a similar roles in circuits, then you'd be like, yeah, I don't care what you call it, that's the cerebellum. And I can't answer that question now.
Really,
but I think
I'm not asking if there is an actual cerebellum. The question is really, to what extent will what you find in C. elegans be extrapolatable to mammals?
Yeah, I mean, if I were, I think, I'm not gonna go to bat for C. elegans having a cerebellum. Perhaps something more like a fly, where it's easier to think about sensory motor transformations and very rapid coordinated movement. That's one of the easier things to study about the cerebellum. I realize it's not the only thing, but that would be a place where I might look for more functional equivalency.
And one other issue I wanted to bring up about the second half of your presentation. I understand the position you're in, and much of what you present looks very appealing, but it's, I think, important to note that the origin of that fortune is associated with a great deal of ethically dubious activity. And that's something that
I'm afraid you and your colleagues need to grapple with.
You know, I work at the Rockefeller University. Is that what you're referring to? Or Carnegie
That
Mellon?
too.
Or any of the great institutions founded by the robber barons, the Koch Cancer Center at MIT. I think we scientists have made our peace with this particular issue. Yeah. Yeah.
There are some significant issues raised.
Yeah. Yeah.
So I would love to get some advice from you, if you can offer it, on getting people in slightly different fields who have some overlapping interests to work well together. We have the Cleveland Brain Health Initiative here. We're trying to get people from all of our campuses, from every department. If you're an engineer and you're interested in brain health, we want to work with you. How do we facilitate that? How are you facilitating that?
It's really hard work to get to work together across fields, especially when people have developed extremely deep expertise in a particular field over long periods of time. And like people think scientists and engineers are practically the same. Like it is not so. It is unso. And kind of developing a common language and a common set of understanding of what the problems are, it takes a lot of time and effort, and it can be pretty frustrating. And I don't think there's any replacement for that. I think this translation layer, which we figured out very quickly was going to be necessary for people like me to talk to people like software engineers, you know, that's one answer, is that there is this sort of level of interpretation. And it was, you know, and it's something that I think maybe also, you know, people get really, they get very immersed in their area, and they get kind of proud of it, and they forget what other areas have to offer them. So I think being humble about that and leaving the door open that we might learn something really valuable from another group is part of that as well. We're doing, in our policy group, we want to work more closely with, there are subsets of patient groups, particularly in rare diseases, who get very involved in the science and in participation and even setting scientific questions for further study. And we're very interested in kind of bringing that group of people into the conversation as well. There are some great examples in my field. Nancy Wexler and her family's work at the Hereditary Disease Foundation was directly responsible for the identification of the gene whose mutation caused Huntington's disease, and because of their own family's experience. And I can also say from that, you know, they brought an amazing group of people into the field, including this, who had never thought about the disease, including this geeky assistant professor from Michigan named Francis Collins, and they brought a lot of moral authority into the field and got people to share their results and work together when people were not interested in doing that in human genetics. And so I think, you know, that there are questions and ways you can focus the field through bringing in more of the affected people that could also be helpful. We'll see how that goes too. That could blow up in our face as well. We're open to failure. Yeah, Emmett.
I was going to ask a question about the C. elegans research. Yeah, I noticed that when you describe these two behaviors, the active and the inactive states, actually to switch from one to another, that transition has to be really fast, you know, because it's like you trigger that central program that produces the activity. While the release of this neurotransmitter that you describe is slow, and also you are describing that even they don't connect to each other, they don't connect synaptically, but it's like diffusion of the neurotransmitter, which make the process even slower. So what I'm thinking is that maybe there is this decision-making or change from behavior depend on one particular neuron that is to be identified maybe. And these are just like a epiphenomenon of what really is happening in the circuit. Yeah.
Yeah, I hear you. I'm not positive how that we really know how fast those transitions are. Like if we keep blowing them up, whether we would see more kind of hesitation along the border, for example. And yeah, there could well be sort of command-like neurons within the circuit that have the properties that you want. That's not the way that we're thinking about it at the moment. That is the way that I think we thought about it, a decade or two ago. But now, just based on what the results look like, we think about it more as sort of a circuit with a certain kind of hysteresis that's in one state and then kind of flips into the other state more or less quickly, but that information is actually distributed among multiple neurons in each of those two states. And this particular model also resembles the way these kinds of flip-flop states work in, for example, transcriptional networks in development, where the original idea that there would be one master regulatory gene for one particular cell type has been modified to realize that there's actually these sort of antagonistic assemblies and the muscle specification factors stimulate other muscle specification factors and inhibit adipocyte specification factors and so forth. That's kind of more of a networked effect and less of a single one. They both have elements of correctness to them, but that's sort of it's more helpful for us right now to try to think across rather than just down to one. Mm-hmm.
Thank you for a wonderful presentation scientifically, and the Chan Zuckerberg initiative I think is very exciting, going from basic science research to the STEM education and promoting opportunities, and also the open science. I found that intriguing. You mentioned about having the understanding of the different cells and having that be open access to everyone. Is there anything else you can speak to of how you're going to promote open science?
Well, we're supporting the preprint movement because that enables people to see papers earlier, and because the same papers might ultimately get published on glossy paper behind a paywall, but when they first appear, but if they are available as preprints, then everyone can see them, whether that person is in the US or in a different country or a scientist or a non-scientist. So there's a lot of thinking right now about access to publication, open access to publications as part of open science. We feel like this gives everyone options. So we're kind of supportive of that, although we're not really trying to pick sides in that thing too soon. We're support all of the software that we fund, all of the work that we're funding is being written open source and being posted on GitHub. We are asking the people we fund post their protocols. People are often being funded to support certain kinds of protocol development. We're asking them to post their protocols on open protocol sites like protocols.io. And with respect to intellectual property, we believe that intellectual property that leads to the development of products and that increases the impact of products, and therefore we're in favor of people taking out patents that will ultimately allow hard work to get done. And we want no share of that, so we're not making any requests with respect to ourselves, except that any work that we support must be made freely available for all non-commercial purposes, including pre-commercial use by for-profit companies. So that seems like a reason. It's like, we're here to accelerate science. We think it's great if you want to make a product out of this, but you can't do. There was an egregious case a few years ago where a technology developed by NIH-funded labs was licensed, given an exclusive license to a company that then started sending cease and desist letters to the laboratories that developed it, saying that they couldn't do it without buying this company's really expensive reagents. So we just think that's really wrong. So that's our boundary for where it is, and we're part of this movement. There's lots of different opinions in the open science movement, what the most effective ways are, and so we're working on it. And again, we're willing to be wrong and to change our direction. We're too early on to know that we're right. And I think in topics like this, what right is changes over time too.
Thank you.
I actually think we're going to cut off the questions. Sorry, we have a reception outside, and you could please talk to Dr. Bargmann there. And again, I'd like to thank Cori. It was excellent.